Good morning, everyone, and thank you for extending this quite kind of invitation. Um, I'm here to tell you how GBA1 variants disrupt lysosomal and mitochondrial homeostasis in Parkinson's disease. I don't really have to give an introduction about GBA1 and Parkinson's in here, but just to give you a background of why and how we studied this. Variants in GBA1 are among the most common risk factors for Parkinson's disease, right? But we still don't know how the G case leads to neurodegeneration. And honestly, I don't know if that will answer, if my talk will answer that still. Anyway, for our work, we use two variants in our study, the E326K and N370S, which lie on the opposite ends of the GBA1 risk spectrum. And we studied these variants in both patient-derived fibroblasts and iPSC dopaminergic neurons. While we did this, we also found similar results in both fibroblasts and dopaminergic neurons. So you'll end up seeing me referring to either of the cell types across my talk. What we basically asked using these cells was what happens to lysosomal function, what happens to the mitochondria, and how is mitophagy affected. So looking into lysosomes, Alessia, my technician at the time, she stained the iPSC dopaminergic neurons with lysosensor yellow-blue. It's basically a ratiometric dye which fluoresces blue, and as it goes into a lysosomal compartment, it ends up becoming, or as it goes into more acidic compartments, the fluorescence shifts from blue to yellow. And we quantified it. We found that the GBA1 variants had reduced lysosomal acidity, right? So when Alicia, my colleague, calibrated the pH, we found that the control fibroblasts had a pH of about 4.7-ish, but the mutants had about 4.8 to 5. This indicates that, of course, there was impaired lysosomal acidification in the GBA1 variants. And when Alicia also stained the cells with DQ-red BSA, she found reduced DQ-red fluorescence indicating impaired lysosomal proteolytic activity as well. What Jana did next was stain the dopaminergic neurons with TMRM and asked what happens to the mitochondrial membrane potential. And we saw that both the GBA1 variants had significantly decreased mitochondrial membrane potential. And this decrease was also accompanied by an increase in the mitochondrial fragmentation. So to study this further, we did electron microscopy, and there was not much difference in the number of mitochondria per field, but you can see that the white arrows there in the controls have more structured mitochondria, and which have more defined cristae and stuff. But then when you observe the black arrows in both the control and the mutant neurons, you will see that either the mitochondria are extremely swollen with very damaged insides, or they're completely hollow in some cases. So when we quantified this, we saw that there is decreased cristae density in these GBA1 variants, along with swelling or fragmentation, as we observed before. Moving on, we also did mitochondrial respiration, and in the whole dopaminergic neurons, we saw there were reduced basal respiration in these neurons. And if you've ever worked with dopaminergic neurons, you know that they are a pain because they end up actually clubbing and forming a ball with as cells as they age. In fact, even before the age, actually. And it gets very difficult to get them to stick to the seahorse plate. And so what Krita did was he isolated mitochondria, and he gave them complex specific substrates, and then did a seahorse. And we could see that there was reduced ADP-stimulated ATP production, indicating that, again, mitochondrial respiration is also hampered in these cells. So one of the most logical explanation we could think of as a connection between lysosomes and mitochondria is autophagy, and in this case, mitophagy. So to study mitophagy, we probed the cells with this ratiometric probe, Mitochema, where the mitochondria, when they're present in a more neutral pH, are green in color, and as they go into acidic compartments, they fluoresce red. And you can see that, in the estrogenic controls, there is quite a bit of red fluorescence, but in the E326K and N370S variants, there is reduced red fluorescence indicating, decreased mitophagy or impaired mitophagy. But in the case of mitochema, this can be explained in two ways. It could either be because the number of mitochondria that are entering the acidic compartments are lesser, or it could also be because our lysosomes by themselves have reduced acidity, right? So maybe that's the reason we don't see a lot of red fluorescence. And so to confirm or to see what kind of mitophagy is it, we stained the GBA1 fibroblasts with citrate synthase, a mitochondrial marker, along with LAMP1, which indicates lysosomes, and LC3, an autophagosomal marker. The yellow arrows out there indicate mitochondria that were co-localized with LC3 to form mitophagosomes, and the white arrows are more mitolysosomes, the mitochondria within the lysosomal compartments. So when we quantified, we could see there was no significant difference between the control and mutants with the mitophagosomal compartments, but there was significant increase in the mitolysosomal accumulation in the GBA1 variants. So this basically means mitochondria and lysosomal contacts persist, but then there is no way for them to clear. And so for us, it's more of a lysosomal problem and less of a mitochondrial problem in this case. And so we sort of further looked at what happens to the whole cell lysates and performed Western blotting. When we quantify, you can see that there was increased phospho-mTOR ratio along with the increase in LAMP1, which further confirms that it is. And there was not much of a difference between the other autophagy markers in the GBA1 variants. So we sort of could confirm that mTOR is hyperactive and there is an accumulation of lysosomes and increase in LAMP1 in these cells. So to summarize the first part of the talk, GBA1-mediated Parkinson's have a barrage of dysfunctions in the form of lysosomes, where they have impaired lysosomal acidity, impaired proteolytic activity, accumulation of lysosomes and autolysosomes, and all this could or could not be because of hyperactive mTORC1. And in the mitochondria, there was decreased mitochondrial membrane potential, reduced oxygen consumption, increased fragmentation, and all of this was also accompanied by decrease in mitophagy. So because it looked more like an autophagy problem or a mitophagy problem, we sort of went about it backwards, and we tried several autophagy inducers, and one of them was rapamycin, which we all know. It is an FDA, it's sort of an immunosuppressant drug used in the market, and it is an mTORC1 inhibitor, the most common one out there. What Alicia found when she treated rapamycin at 200 nanomolar overnight is it was capable of improving the autophagy and mitophagy, but it also was able to improve the mitochondrial membrane potential, and it reduced the mitochondrial fragmentation that we found in the GBA1 variants. So we first tested our inducers in fibroblasts and then took them into dopaminergic neurons. Right, so we then confirmed this, and indeed rapamycin was able to rescue mitophagy and mitochondrial membrane potential along with the lysosomal acidity, right? Now, all our results till now tell that it could probably be a mTORC1 problem, and it could be a lysosomal problem. And our literature search said that there is indeed a connection between mTORC1 and VATPase, so what is VATPase? It is the vascular ATPase, which is important in maintaining lysosomal acidity. And this VATPase is sort of made up of two subunits, the V1 and the V0 subunit. And unless the V1 and the V0 subunits come together, there is no proton exchange that happens. And under amino acid rich conditions, this the mTORC1 sits on the lysosomal membrane and prevents the formation of the functional VATPase complex formation and hampers or does not allow proton exchange to happen. But under amino acid starvation conditions, mTOR sort of gets displaced from the lysosomal membrane and then moves into the cytosol and allows VATPase complex formation to happen. Now this scenario, it completely happens in terms of metabolic, in terms of a metabolic scenario where it's under the conditions of amino acid starvation and repletion. We wanted to see if this is the same case even in our GBA1 cells. So we first looked into the cellular lysates, whole cell lysates of the GBA1 variants along with the controls. And we found that there was indeed accumulation of LAMP1, but then there is no difference in the ATP V1 or the V0 subunit levels in the cells. But when we isolated the lysosomes and when we asked the same question, we saw that there was an increase in phospho-mTOR levels on the lysosomes, along with an increase in LAMP1 levels, but there was also a decrease in the V1A and V1H, or the ATP V1 complex levels, but there was not much difference in the ATP V0. Now, when we treated the cells with rapamycin and did the same isolations, the increase in phospho-mTOR that we saw was decreased in the variants, and along with that, the V1 subunits were also, were rescued to that of the control levels. So essentially, ATP6 V1 complex levels are reduced on the lysosomes, and mTOR is capable of rescuing it, right? But to confirm this more, what we did was we basically performed fluorescence lifetime imaging using FRET probes which sort of looks into the interaction between V0 and V1. And when we look at the lifetime of V1, which is the mobile complex from the cytosol onto the lysosomes, and when we measured the average fluorescence lifetime, we saw that in the controls, the lifetime of the V1, B2 was around 600 picoseconds. But when you sort of look into the mutants, both N370S and E36K had a lifetime of about 750 to 800 to 850 picoseconds or so. So this confirms that the VATPase complex is indeed dysregulated, and rapamycin is able to restore it. But talking about rapamycin and mTOR is like opening a Pandora's box. There's, rapamycin is a jack of all trades, and so is mTORC1. So it gets very difficult to pinpoint on what exactly is the mechanism. So to confirm if it is indeed something to do with the pH, we collaborated with Orion Shirihai and Mark Grinstaff and got acidic nanoparticles. Now these nanoparticles are fluorinated nanoparticles which have a greater selectivity and which are relatively stable at physiological pH, but when they are taken up into the lysosomes, they are, they sort of get activated and they're more stable even in an acidic compartment. So when we tested this, what Sylvia did was she confirmed that indeed these nanoparticles are specific to the lysosomes, and she labeled the rhodomain nanoparticles along with lysotracker blue, and there was not much difference in control or mutants taking up these nanoparticles. And when Alicia calibrated this pH, she found that the increased pH that we saw in the GBA1 variants was now reduced to about 4.6 to 4.8-ish, right? And what we also saw was, Alessia saw that, once 180 micrograms of acidic nanoparticles was capable of rescuing the lysosomal acidity and the mitochondrial membrane potential, along with mitophagy in these, in the iPSC dopaminergic neurons and the fibroblasts. Now, we also wanted to ask the question if, the acidic nanoparticles have an effect on the VATPase complex itself. And so when we performed the FLIM, we saw that instead of decreasing the fluorescence lifetime, acidic nanoparticles were actually increasing them in the control fibroblasts. And there was not much change in the GBA1 mutants, probably indicating that, not probably, but indicating that acidic nanoparticles are acting independent of the V8PAS complex, and they're able to acidify it artificially. So as a summary, GBA1-mediated Parkinson's has both lysosomal and mitochondrial dysfunction, possibly because of the hyperactive mTOR, which then leads to incomplete V8PAS assembly, and then all the downstream dysfunctions of lysosomal proteolytic activity, dysfunctional mitochondria, and dysfunctional mitophagy. And when we correct the lysosomal pH, the lysosomal acidity is restored, which also restores the proteolytic and the hydrolase functions, and it sort of corrects the autophagic and the mitophagic function. And because of this, there is sort of a healthy mitochondria that we see in the cells. And as a take-home, lysosomal acidification appears to be sort of the upstream lever of all the problems that we are seeing in the mitochondria and autophagy. And when we correct them, then reacidifying this lysosomal pH is able to rescue the pathophysiology in these cells. That's my thanks. Thank you mainly to the donors and the patients for the cells that we are able to work with. Thank you to GBA1. I'm coming back after 1 year. I came for the first meeting, and this is my second one. Thank you for the invitation, and that's the Duchenne lab. That's Michael Duchenne out there at the left end. Thanks to Michael J. Fox, Parkinson's, ARUK, and everyone else, and UCL, where I actually did the work. I'm currently based out of Cardiff. I work with Caleb Weber. Our question till date was never, tells you about how GBA1 problem exists in both fibroblasts and dopaminergic neurons, and that still sort of opens up a big question: what happens, and why is it only affecting the dopaminergic neurons, and why does it lead to Parkinson's? Which I hope to answer maybe in a story for another day, which we are sort of working on now based on the recent results that we've sort of got in the Weber lab. So yeah, thank you.